and welcome to part 4 of our look at processes involved in cell biology. This video is going to be very dense. It's probably going to be a little bit longer than some of the other ones because the process that we're looking at today is protein synthesis. And exactly how do we go from these amino acid structures to having them linked together to create these primary proteins that can then become secondary proteins, tertiary proteins, and quaternary proteins. There is a story to be told when we are looking at all of this, and so that is where we are going today. To start this conversation off, what we need to understand is that the idea of protein synthesis is a foundational belief system based on research and data and experimentation that we've come to understand within biology. So when we talk about something being a central dogma, we're talking about it being a foundational concept for which everything else is built upon this understanding. All right, so much to the point that you look at evolution. Evolution is rooted in this central dogma of biology that we're about ready to go over, this foundational understanding. You look at things such as vaccines and how we produce vaccines. You look at replication. You look at mutation rates. You look at response to environments. All of these things are all predicated on having this foundational belief and understanding that we call the central dogma of biology. And this foundational understanding is very simplistic. DNA leads to RNA, and RNA leads to proteins. It is this linear progression of going from stored genetic material on the genes, extrapolating out that information, converting it to an RNA-based language, and then using that RNA to go ahead and create proteins. This is different than what we saw when we looked at DNA replication. DNA replication was replicating the entire chromosome. But when we talk about protein synthesis, we're talking about only replicating specific regions of the chromosomes. And those specific regions are what we define as genes. Specific regions found on DNA is what we define as being genes. Genes code for a diverse group of proteins. So the information used to create the proteins are stored within these genes. Now, the genes, you can have variety within the genes. So, variation within the genes is what we refer to as an allele. So an allele is a variety or a variation of the gene. Do you have brown eyes or blue eyes? If you're a pea plant, are you tall or are you short? Do you have a widow's peak or do you lack a widow's peak? Are your earlobes attached or are your earlobes unattached? Do you have a straight thumb or do you have a hitchhiker's thumb? They're all alleles. All right. Do you have a hitchhiker's thumb or a straight thumb? Right? So in other words, when you hold your thumb up to give a thumbs up, does your thumb curve way back, which is a hitchhiker's thumb, or is your thumb straight up and down? Right? The gene would be thumb position. The alleles would be hitchhiker thumb or regular thumb. Right? If you're a pea plant, the gene would be plant height. The alleles would be tall or short. So that information is encoded in the DNA. We then take that information and through a process that we're going to talk about here in a few minutes, we create an RNA copy. And then that RNA copy is sent off into the cytosol of the cell to make proteins. All right? That simple equation is foundational to everything in biology. Because if you get a change in the gene, you change the allele, and if you change the allele, you alter the protein. And when you alter the protein, sometimes nothing happens. Sometimes you get a mutation that leads to a disease or a condition. Sometimes you have greater implications that lead to evolution. How an antibiotic works is rooted in understanding the central dogma of biology. How viruses are able to replicate using your own machinery is rooted in the central dogma of biology. Whether or not you are at a higher risk of alcoholism, whether or not you are at a higher risk of heart disease, whether or not you are at a higher risk of developing some kind of osteoarthritis, or lung cancer, or stomach cancer, or anal cancer, or ovarian cancer, is rooted in this understanding of biology. And the beauty of this is it doesn't happen any other way. DNA to RNA to proteins. But remember, the first rule in biology is there's an exception to every rule. And there is exception to the rule. All right? So in other words, and this isn't an exception to the rule, you already know this. At least you better know this. DNA can make DNA. Right? We already know that happens during S phase of interphase. We refer to that as DNA replication. All right? This is a no-brainer. All right? And then from there, DNA to RNA to proteins. All right? And we refer to that as being protein synthesis, this thing that we're going to be getting into here in a few seconds. But there are exceptions to the rule. Viruses are an exception to the rule. Now, viruses are not living. So they're allowed to break all of the rules because they're not living. They don't play by the same rules to begin with. But what viruses can do is you can have RNA viruses. COVID-19, right? SARS-2 COVID. COVID-19 is an RNA virus. It is a positive single-stranded RNA virus. What does that mean? What that means is its genetic material is based in RNA. It's a single strand of RNA, and because it's positive sense, it has a higher resistance to evolution. COVID-19, since December, is estimated to have had about 21,000, no, I lied, reverse that, has had about 12,000 detected mutations within its genome since November or December of 2019. At the time of this video filming, we are in September 2020. And so over that 10-month span, the virus has been able to replicate enough times to accumulate 12,000 mutations. And remember, because it's positive sense, it's actually guarded against. The rate of mutation is a lot slower for other reasons that we're not going to get into. Well, how do you get those mutations? Well, you get those mutations because as an RNA virus, you insert yourself into a host, and you actually inject your RNA into the nucleus of the host cell, and that RNA then basically secretes enzymes that converts the RNA into DNA, and that DNA is then assimilated into your genome. So the next time you engage in DNA replication during S phase of interphase, you replicate the viral proteins. And when you make the viral proteins, you make the viral parts. And then your cell actually then goes ahead and puts the parts together. So the virus doesn't do anything to replicate itself. Except infect the host cell. That's it. Once it's in, you do the rest of the work. Think about the irony in that. All right. So RNA viruses can actually create DNA or can actually lead to the synthesis of DNA versions of the RNA. RNA viruses can go ahead and create more RNA of itself. So they violate this rule every which way possible that you can think of. Now remember, DNA is only made up of 4 base pairs: adenine, thymine, guanine, and cytosine. And so the entire genome, every single gene, is made up of only 4 base pairs. And it's the order of those 4 base pairs that are then broken down into smaller subunits that gives us the RNA to produce every single protein. Tens of thousands of different proteins. And the process that we employ to go ahead and create all of those proteins by going in and first synthesizing a gene within the DNA, and creating an RNA copy of that gene, and allowing that RNA copy to then guide the production of a protein is what we refer to as protein synthesis. And that is where we are heading over the next few slides. And by few slides, I mean 15 slides. So let's get some terminology down, because I'm going to start going through this process, and you need to be able to kind of understand what it is that I am referring to. All right. So when we talk about RNA, there's actually a few different types of RNA that we need to consider. We have messenger RNA. The M in a messenger, or I'm sorry, you have mRNA. I told you the answer. And the M in mRNA stands for messenger. Right, so mRNA is messenger RNA. It's the messenger RNA that carries the DNA instructions to the site of protein synthesis. We then have rRNA, right, where the R stands for ribosomes. The R stands for ribosomes. And it is at the ribosome where messenger RNA is read to produce a protein. So rRNA, or ribosomal RNA, is actually the site of protein synthesis. Now you should start putting things together. Right? You should start putting things together. What should you start put together? Well, you should start to put together um, that ribosome word. Rough endoplasmic reticulum is studded with ribosomes. Ribosomal RNA is the site of protein synthesis. That is happening on the rough ER. And so all of those little ribosomal studs that are lining the endoplasmic reticulum is really our RNA. And then we have tRNA, where the T stands for transfer. And it's the job of the transfer RNA to bring the appropriate amino acid to be linked together based on the messenger RNA that is being read by the rRNA. I'll say that again. It is the job of tRNA, transfer RNA, to bring the appropriate amino acid to be linked together based on the messenger RNA code by which the rRNA, or ribosomal RNA, is reading. Now, it's also in rRNA, or I should say it's also in messenger RNA, where we see any place where there would be a T, it is going to be replaced with a U for uracil. So wherever there is a T that would be in rRNA, that is going to be swapped out with U for uracil. So let's start to look at this process. Let's try to get this this process down a little bit in theory. So protein synthesis is broken up into two distinct phases. Right, so protein synthesis is broken down into two distinct processes: transcription and translation. All right. Transcription takes place within the nucleus. Transcription takes place within the nucleus, and it involves unzipping the gene and copying the DNA, making up that gene, into messenger RNA. Into messenger RNA. So we are going from DNA to messenger RNA. And that is happening during transcription in the nucleus. Then, in the cytosol, at the rRNA, we have a process called translation. And translation involves reading or interpreting the messenger RNA to be able to create a linear strand of amino acids that you all know as a primary protein. So at the end of this process, we would have created a primary protein, a linear strand of amino acids that have been linked together through the formation of peptide bonds. And we're going to find that through the process of protein synthesis, how we create those peptide bonds is different. It's not just a matter of just doing dehydration synthesis. There's a little twist involved with this. This diagram here just kind of shows you that overview. All right, so you can see here where transcription has taken place. All right, we are going to make an RNA copy. We're going to talk a little bit about this on the next slide. I'm going to draw this out for you a little bit more. There's an intermediary step. There's what we refer to as pre-messenger RNA before the actual messenger RNA is created. But notice all of this is happening here in the nucleus. And then that messenger RNA leaves the nucleus and it goes to the site of the ribosome. And that is where translation is going to take place. Transcription is in the nucleus. Translation is in the cytosol. All right. And by the way, the DNA does not leave the nucleus. And remember, RNA is less stable. Maybe this is starting to make a little bit more sense on why RNA is less stable. If it's not, we'll get there. We will get there. All right, let's look at this process. So we have I'm going to draw this out. God help us as best as I can. All right, so this is the nucleus. Right, these little gaps that you see in there, those little gaps are nuclear pores that are within the nuclear membrane. All right, so this is the nuclear, let me just rephrase that, this is the nuclear membrane. Right there, right? And then there's little gaps there are the nuclear pore. Inside of the nucleus, there is DNA. Right, so here's our DNA. Now, what I'm not going to bore you with is the role and the function of things like RNA polymerase and DNA polymerase and helicase and ligase, because all those characters are still at play here. Right? And in fact, if we added one, RNA polymerase, these guys are all still at play. We're not gonna go there. Not yet. Right? Not yet. But what'll happen is those enzymes are driving this process, and they identify the gene that needs to be made. And so here's the gene that needs to be made, right there between my two gray lines. Only that area is unzipped. And when that area is unzipped, what we define as the leading strand of genetic material, the leading strand is what is going to be replicated. And so this strand, I'm just going to go ahead and say, is the leading strand, and its DNA base sequence for that gene is as follows. All right, so there's my sequence. All right, there's my sequence. All right. TAC, GCA, TTC. Now, don't be confused. All right, because this is my DNA, so it's allowed to have T's in it. All right, my DNA is allowed to have T's in it. Now, I'm going to go ahead and I'm going to create a messenger RNA copy of this. So where I see a T, I'm going to have an A. Where I have an A, because I'm RNA, I'm going to have a U. C is going to be a G. G is going to be a C. G, U, A, G. All right. So there is my messenger RNA copy of the DNA. But remember, this is pre-messenger RNA. Because what this is actually made up of, pre-RNA is made up, it's made up of areas that we call introns and exons. Introns are non-protein coding areas of pre-messenger RNA. All right, so that's what the introns are. Exons are coding regions. And so what ends up happening is our pre-mRNA actually goes through and gets sliced up. The introns are removed. And then the remaining exons are squeezed together. And it's this leftover exons that have now been squeezed together that will create the final version of messenger RNA. So we're going to pretend that that sequence that I have up there is still legit. All right, we're going to pretend that that is the final messenger RNA. So messenger RNA, that sequence is AUG, CGU, AAG. Right? That is my messenger RNA. So again, pre-RNA, pre-messenger RNA, it's made up of these areas of introns and exons. Introns are non-coded regions, so they actually get removed. Exons are left over. That's what's actually coding for the protein, and they get squeezed together. But there's something else that happens here. All right. The other thing that happens here is that the cell, the nucleus, can actually take those exons and rearrange their order. And by rearranging their order, they create multiple types of proteins from just one gene. They can reorder the sequence and the order of the exons and create multiple types of proteins out of that same exact sequence. Once the exons have been rearranged and then squeezed together, we take the messenger RNA and it leaves the nucleus. And when it leaves the nucleus, it goes to the rRNA. Which is what that thing is here. Right? This is the rRNA. Now again, that is in the cytoplasm. All right, that is in the cytoplasm. So the other thing that I will mention is that to help guide so that way the cell knows exactly where this protein starts, it looks for a start sequence on the DNA. And the start sequence basically says when to start creating the messenger RNA copy. All right? And that start sequence is what we call a TATA sequence. TA, TA, TA. Right? Or sometimes it is a TA, TA, A. I think there's one more A at the end of that. Right? Either one of those two sequences of T's and A's will give you the go or the green light to begin producing that messenger RNA. So far, all of this, everything happening in the nucleus, is what we're defining as transcription. And that is exactly what this shows. Right? So this kind of gives you a sample of practicing creating messenger RNA. That's the DNA copy. Right? And so A would become U, T would become A, G would become C, C, A becomes U, G, C, G, U, A. And so this is the messenger RNA, while the top strand is the DNA. All right. Okay, I think I'm going to break this up into two videos, just because it is a lot of content and detail. So we're going to stop here, and the next video we'll pick back up with translation. What happens now during translation? So we're halfway through the process of protein synthesis. It's a good place to start. And with that, I'll catch you on the flip side.